Okay, so in Unit 4, this organic chemistry study of compounds was one of the things that is on the list to study. So it's the definitions in that section that you want. In there, the section on naming here, so how this works with the carbons was one of the sections in there. This section on the isomers. And when there was the chemical groups and structures, it's this section here, right here, the chemical group and the structure. I didn't put in any of the stuff from these over here, because that's kind of one of those things that in just working with the stuff over time, you tend to, the information comes together from using it over time, not necessarily from memorizing it from a table. So with the macromolecules, these are going to be your large biological molecules that we're looking at this point. And there's 4 main classes that these are going to fall into. So you've got the carbohydrates, lipids, proteins, and nucleic acids. And their architecture, their shape is going to explain how they work. So we're going to be looking at each of those. One of the things you're going to see is a lot of them are polymers, which is going to be a long molecule that has many similar identical building blocks linked covalently. You could kind of think of them being like Legos, where you got lots of similar pieces that get stacked on to make your bigger structure in there. And that's what you're going to find with these macromolecules is most of them are going to end up being polymers. The monomers are the repeating units of the smaller molecules that will serve as the building blocks, and they have their own functions, which a lot of times the accumulation of all of those different functions together makes the bigger overall piece in there. So a lot of these are going to be made through what we call a condensation reaction, where you've got 2 molecules that are going to covalently bond together. And when they do, in the process of their bonding, now you've got more atoms sharing electrons. So when you start sharing with somebody else instead, you have to let something go. And what they let go is a water molecule will be pulled off. And so it's a dehydration reaction in there. So one of the things when you consume food and you build things, you actually get what we call metabolic water out of the process. When you take the ingredients of your food, the compounds that it's made out of, and reuse them to build other things, a lot of times when you glue the pieces together, you pull off the water in the process. So you have more water intake than you usually anticipate just from your food in there. Enzymes are going to be important. These are going to be the macromolecules that are going to help speed up the chemical reactions. They do a lot to help control the chemical reactions. A lot of times it won't take place without the enzyme present. You can kind of think of it as where you maybe have 2 things that get really close, but you need to have something in there that's kind of a piece in between that helps pull them together temporarily so they can reach each other and bond. That's what an enzyme is going to do, is help them be able to make that last little step for their interaction in there. So hydrolysis, this is going to be when we break a polymer down into its monomers in there. Yeah. Oh, there we go. That's doing it. And so with that, you have to add water in there. And that's part of what we do with digesting food, is you actually have to have some water in order to be able to break down some of these nutrients that you consume. So the first group we look at are the carbohydrates. So these are going to include sugars. So a lot of times people, they'll have different impressions that come through your mind in the everyday world of a carbohydrate versus a sugar in there. In chemistry, they're the same thing in there. The sugar is a common name for the carbohydrate. And the polymers of sugars are going to be your more complex ones. So you have monosaccharides. Saccharide also means sugar, which is going to be one. Mono means one. Disaccharides are going to be two sugars hooked together. And then polysaccharides are where you have many sugars hooked together. So when we look at the monosaccharides, these are going to be your single sugars. And they're all going to be multiples of CH2O. So if you take a look at, for example, glucose has 6 carbons in there. You would have 6 carbons, 12 hydrogens, 6 oxygens, because you just multiply that whole unit and everything in it by 6. So that's the basic ratio that you have in there. And it's going to have a carbonyl group on there and multiple hydroxyl groups. So some of the monosaccharides will make aldoses or ketoses. Their names are going to depend on the size of the carbon skeleton. So hexoses are ones that have 6 carbons in there. A pentose has 5, and a triose has 3. Those are the 3 of them that you'll run, 3 groups you'll run into the most commonly in there. So their spatial arrangement around the carbon will differ in there. Sometimes you'll see them represented as little chains. Other times you'll see them represented as more of a ring structure in there. So what do we use these for? These are a major nutrient for energy for the cells. It's also used as a building block to make other macromolecules, but this is the one that your cell wants to use to get energy. So a disaccharide is just going to be 2 monosaccharides, and they're joined by a glycoside linkage. So that's what they call the specific dehydration reaction between the 2 monosaccharides. You will want to know what makes up these disaccharides. They're something that you'll run into a lot. I believe you guys have to take nutrition in nursing, and so your disaccharides and monosaccharides come into play in there as well. So you might as well learn them now. So maltose in there, or malt sugar, is actually 2 glucoses linked together. Lactose, your milk sugar, is going to be a glucose and a galactose hooked together. And then sucrose, which is your table sugar, is going to be fructose and glucose hooked together in there. And those are all ones that you will run into in the food industry, as well as see really commonly in biology. So the polysaccharides, this is going to be multiple monosaccharides joined together by those glycoside linkages. Oh. Next time the typo gets to stay. So these are going to be used more for storage or for building materials. If you need to get energy, you break these down into the monosaccharides and disaccharides, and those feed into the process of supplying energy in there. So starch is a glucose polymer that's found in plants with plastids. It includes the chloroplasts in there. So any idea of something you might consume that's going to be high in starch? A plant. Potatoes are a good example in there with the starch in there. So like the food industry, some people go, well, I ate my vegetables, I ate my potatoes. Well, technically they are a plant. They are not necessarily considered a vegetable food like that. They're more of a starch food because that's primarily what they have in there. Glycogen is going to be the polymer of glucose that animals store in their muscles. So when you exercise, one of the things you're going to tap into is the glycogen stores in your muscles, and you will be able to break those down very easily in there to be able to get energy out of them. So the starch linkages in there, what's going to differ in these is how we hook them together. When they're hooked together differently, you have to have different enzymes to break them down. So the starches are going to be joined by 1,4 linkages, which means carbon 1 is attached to carbon 4. And so the example with that would be maltose. And the angle at which they join with that makes them helical shaped in there. So amylose is a simple starch. It's unbranched in there. And then amylopectin is going to be branched with 1,6 linkages. Here's another one of those places where there is not supposed to be a 3 in there. The formatting kind of took over on there. And then cellulose, this is a major component of tough cell walls in plants. You have 2 rings that form glucose, the alpha and beta in there. The starch is alpha types of bonds in there. The cellulose is going to be all beta bonds, and it's going to be straight and unbranched in there. So what's going to make the difference between what you can digest and what you can't digest is you can digest the alpha bonds that starches make. You cannot digest the beta bonds that cellulose makes. So if you go eat grass, which is going to be high in cellulose, grass is going to come out the other end with the only change being how much you chewed the grass, where a cow actually has enzymes to break that down in there. So that's how you end up with some of those things like corn that just don't digest in there. So when we talk about insoluble fiber, it's mainly cellulose. And you go, well, why would you eat something that you can't digest and break down? The fiber does help to keep things moving in the digestive system, keep things from building up in there quite as much. So it does play an important role in the health of the digestive system. And then we've got one more polymer in there, chitin, which is a polysaccharide that's in arthropod exoskeletons and a lot of fungi in there. And you can actually break this down. So you can eat mushrooms and you have no problem breaking that down. And while it may not sound real appealing to a lot of people, you can eat the insects too. Shells and all. So there are, some cultures do eat like shrimp. They'll eat the entire shrimp or the heads in there. So if you go to some sushi places, you can get shrimp heads that are fried in there. And, if you've never tried it before, it can be, if you didn't grow up eating that thing, it can be a little disturbing that there's these eyeballs under the deep fried stuff. But I tried it. It actually wasn't bad. They're a little sharp, sticking out in your mouth, but you have no problem digesting the chitin in there. So the lipids, these are all hydrophobic, which means they're not going to like water. It will include waxes, certain pigments, fats, phospholipids, and sterols. So fat is constructed of glycerol and fatty acids. Glycerol is a 3-carbon sugar, and to each of those carbons in there, you're going to have a fatty acid attached to it. So it's kind of a structure like this. And so they're commonly called a triglyceride in there or triglycerol. You got 3 fatty acids attached to the glycerol in there. When we talk about a saturated fat, it means that you've got no double bonds between the carbons. So in these long chains of fatty acids, they're usually going to be 16 to 18 carbons in a human. All of them just attach to the neighboring carbon once in a saturated fat in there. So it's attached to as many hydrogens as possible. With the unsaturated fats, these are going to have one or more carbon-carbon double bond in there. So it will share with one of the neighboring carbons twice. If it's monounsaturated, it does that once. Your most common source of monounsaturated fats in a human diet is going to be olive oil. If it's polyunsaturated, it will do that more than once. It's usually twice that we talk about in the human diet in there. So those would be your polyunsaturated fats like vegetable oil, canola oil, cottonseed oil, a lot of those types of things. So we know with the carbon-carbon double bonds in there, the more of those you have, the less stable it is, the more liquid it's going to be at colder temperatures in there. So one of the things that has happened is we've figured out ways to hydrogenate oils. You take an unsaturated fat and synthetically convert it to a saturated fat. And one of the things they found with doing this is that you could make these weird little fats that don't separate out of food. So one easy way to see it is if you get peanut butter that is just like Adam's peanut butter, or you get it that's just peanuts and maybe salt in there. When it sits for a while, it separates. You have the oil separating from the mixture in there. But some people like to have peanut butter that it's all blended together and it doesn't separate. And the way they keep it from not separating in there is to add these hydrogenated fats in there. And so I look at that and I try to avoid those hydrogenated fats as much as possible. So that in my book is dog peanut butter that you give dogs pills with. But that is the reason why it doesn't separate, is it's an artificial thing in there. So margarine is an example of that as well. And for a long time, these things were being promoted as being a healthier option. And what we know now is that the body doesn't really break those down the same. When we make them, normally you're going to have a combination of some cis and trans fats that will occur in nature. But when you make them artificially, you get a higher percentage of them that are these trans fats. So where we talked about cis, meaning the legs were next to each other, trans, they're across like that, it takes different enzymes to break those down. And it actually alters the cell membranes. They don't break down in the bloodstream the same. So they're finding that there's actually increased risk for heart disease with more of these trans fats, even though they were originally, people thought they were going to be better for their health to have the margarine and things like that. You're actually better off to go with the real saturated fat if you're going to eat that your body knows how to deal with than the trans fats. So now you're starting to see it being mentioned on labels that they're free of trans fats and things as people are becoming more aware of that. So the phospholipids, this is going to be two fatty acids attached to a glycerol, and the third one is going to be joined to a phosphate group. And we have these in our cell membranes. So here, instead of it being the fatty acid, we put a phosphate on there. And what these look like more is you've got this phosphate head and these fatty acid tails that hang down from it in there. Where you're going to find these is in cell membranes. What makes them so good in cell membranes is where you have the phosphate actually has a slight charge to it. It's going to like water. Down here you've got these fatty acid tails. They don't like water in there. So it gives it a unique property in that one side of it is going to be happy to be against a watery surface. This side does not want to be against a watery surface. So as a cell membrane, it's going to keep the watery in the cell, and you're going to have another one of these that is going to face the opposite direction. It's going to keep the watery environment that's outside the cell outside, because neither one of them want to cross through this zone where they're not wanted in there. And then we have sterols. These are going to be lipids that have a carbon skeleton ring, or a carbon skeleton that consists of 4 fused rings in there. And then they get different chemical groups attached to them. These are found in a lot of the hormones. When we call it a steroid hormone, it's because it's derived from a sterol. And cholesterol is just the most common of those. It's found in animal cells. There is no cholesterol in plant cells at all. So it may contribute to atherosclerosis with the trans fats and saturated fats. So one of the things when they're looking at your risk of cardiovascular disease, they look at is your cholesterol and your total blood lipids in there. And then we have the proteins. This is going to make up a lot of structures in there. So when you see protein, you want to think this is a structural molecule. It's not meant to be one for energy. Consuming protein actually really does not do a lot to give you much energy. It is mostly used for making structures in your body. When you put it in there and it has to be used for energy, it's not going to be terribly efficient at doing it. And there's some places like your brain that just really do not like to run on that at all. So if a person is on a high protein diet in there with very little carbohydrates, one of the things they'll find is difficulty concentrating and remembering things. The people around them will find they might be in grumpier moods and things like that. Usually they don't own up to it themselves, but if the brain is needing more carbohydrate in there, it will affect how it functions in there. So things we use the proteins for to make enzymes, structures, storage, transporting things, hormones, receptors, and contractile and motor tissues, and then defensive things. So as polymers, they're all constructed from the same 20 amino acids. So in humans, we have 20 different amino acids that are found in there. There are a few other amino acids out there, but in humans and animals, you find the same ones. And a protein is going to be one or more of these polypeptides. So sometimes you'll see that amino acid being referred to as a peptide in there. What the amino acids are going to have is a carbon in the center. And then you're going to have an amine group on one side, an acid group on one side, a hydrogen, and then this R that's a variable group. So they get the name amino acid because they've got the amine group and the acid group on there, their functional groups. So this R is the only thing that's going to be different in all 20 of the amino acids. This whole part is always going to be the same. This is the only thing that's going to have a different functional group there. And you may look at that and go, well, we only had 7 functional groups. There are more functional groups out there than what is in your chart. And they can be combinations. It won't necessarily be just the functional group alone. You can have a couple of different functional groups on there. So it's an asymmetrical alpha carbon in the middle. So when we go back to those isomers where you have that asymmetrical carbon in there, and these things are different, they're going to be the right-handed and the left-handed molecules. It has to do with how it spins light that goes into it. So with amino acids, you'll see this L and R or L and D designation in there. What you find in the body is most of them are in the L conformation. So when you look at amino acids in the health food store, you'll notice they'll have an L in front of them. That's what it's designating, is that rotation in there, which of those isomers it is. So those, anybody remember what we call those isomers that are going to have the orientation like that, the right and the left, or the mirror images? So those are the ones that are the enantiomers in there. So our groups of amino acids in here, we can divide them up different ways in here. So some of these are going to fit into more than one of the groups. And I just listed them on here so that you've got them in a convenient location. So the ones that are non-polar, what that means is they're going to interact more. They're going to be dissolved in fatty substances more, and thereby being non-polar. You've got glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline in there. So don't worry about knowing the names of them. Yeah, because they're nonpolar, they're going to get along with nonpolar molecules, which are fats in there. The polar ones are going to get along more with watery substances, because the rule of thumb is always like dissolves like in there. So you've got serine, threonine, cysteine, tyrosine, asparagine, and glutamine that are polar. Some of them are electrically charged and acidic, and there's aspartic acid and glutamic acid in there. Some of them are electrically charged and basic in there, so lysine, arginine, and histidine. And that's going to be their pH when they're in something. A lot of times we tend to look at it and go, okay, we've got amino acid, that means they're all acids, and they're not always. The name's a little misleading like that. What is usually, outside of the chemistry world, most interesting to people is these last three categories in here. The essential ones are going to be ones that you need to consume through food. So they're ones that your body cannot make from other amino acids. And then the non-essentials are ones that your body can make. And then a lot of times you'll hear bodybuilders or people that work out a lot talking about the branch chain amino acids, the BCAA. These are ones that are metabolized in the muscle rather than the liver. And so somebody who is doing a lot of exercise in there, trying to build more muscle in there, when they're stimulating muscle growth, would want to have more of these amino acids available. So it's always the best idea is to have the wide variety of amino acids, which animal products are going to cover your bases for that. If you eat any animal product, you're going to get all of the amino acids in there. It's just sometimes with certain types of activities and lifestyles, you might want to shift the balance a little bit more. You wouldn't want to consume only branch chain amino acids. There'd be a lot of things you wouldn't have the tools for. But having a higher portion of them would be beneficial in there. So when we link amino acids together, they're linked by a peptide bond. And that's a term you'll see over and over again, the peptide bond in there. It's a covalent bond in there that's formed when the carboxyl group is next to the amine group. What happens is you have one of these hydrogens come off and this OH come off. It makes water, and they hook together in there. So when we make a polypeptide or a protein, it's going to be formed by a unique sequence of amino acids. So you take a sequence of amino acids, they will automatically fold up and go do whatever job they are supposed to. That's what's going to determine what that protein's shape and job is that sequence of amino acids in there. So when we look at what the DNA tells us to build, it gives you instructions to make the sequence of amino acids in that protein. So its function is going to be a result of the 3D architecture in there. So when they fold up, it's going to make a three-dimensional shape. So kind of like you have a claw that fits around something in there. That's going to be how those proteins are going to function. That's pretty much how everything functions in cells in the body, is with this three-dimensional shape where they fit together. It's kind of like fitting square boxes over square objects, round boxes over round objects in there. So when you have your functional protein, it's one or more of the polypeptides in its own unique shape. It will fold up that way spontaneously, and nearly all of its functions depend on its ability to be able to recognize and bind to another molecule like that. So an example would be with antibodies. These are what your body will use to try and protect itself from invaders that will come in, which are going to have proteins on them. And those are called your antigens. That antibody will only bind to that specific invader in there. So if you get vaccinated for, say, hepatitis B in there, you're going to make antibodies that will attach to hepatitis B and absolutely nothing else. It is very specific in there. And that specificity in there actually means that antibodies are a really useful diagnostic tool in there. If you want to look and see, does somebody have antibody towards something, you can take their serum and you can put some of that antigen in there, and when you put the antibody on it, it will actually clump together if that antibody is present in that serum in there. So they're usually going to be either globular or fibrous proteins. The globular ones are roughly spherical in shape. They literally are like a glob. And then the fibrous ones are going to be more of long fibers in there. So your proteins have 4 levels of structure to it. The primary structure is what the unique sequence is of the amino acids. If you change that, you change everything else about it. So once you have that unique sequence made, it's going to form either alpha helices or beta pleated sheets, and that will be its secondary structure. So you're going to have these regions of it that will repeatedly coil up if it makes the alpha helices, or they will repeatedly fold into what they call the beta pleated sheets. And that happens spontaneously in there. So when it's the alpha helices, you've got a hydrogen bond on every 4th amino acid. When there are beta pleated sheets in there, there's 2 or more of the regions lying side by side that get hydrogen bonded together. So the hydrogen bonds are actually really important with your amino acids in helping to hold them into the shapes that they are. Yeah, in any of your proteins in there. So the tertiary shape, this is going to be the overall shape from the interaction of the R groups on there that's superimposed on the secondary structure. So if you kind of think of it as like when you do origami with paper, you make a few folds and then you fold it some more on top of that. That's your paper is your primary sequence in there. Your alpha helices or beta pleated sheets are those first folds that you make. And then the tertiary structure is going to be those additional folds that you make on top of it. So some of the things that you'll create in there, there may be some hydrophobic interactions where you're going to cluster in this little core of area that's out of contact with water in there. Some of them will make these disulfide bridges. So cysteine has these sulfhydryl groups on there. And when you get those close together, those sulfurs can bond to each other, making the disulfide bridges. That's important because those are really strong bonds that will make stronger structures. So any idea what you might have in your body that's going to have a lot of disulfide bridges? Fingernails and hair. It helps give those a lot of strength in there, is having disulfide bridges in there. And then your quaternary structure is the overall structure that results from aggregation of polypeptide subunits. So when you make these polypeptides in there, you've folded up your paper. Maybe you want to make some fancy animal here that takes 2 pieces of paper to make. You make the head with one and the body with the other in there. You can think of that being your overall final structure in there, with it being multiple polypeptides in there. Is that idea making sense, how we have these 4 components of the protein structure? So if you change the sequence of the amino acids, it changes everything else because it won't fold up the same way. So when we denature a protein, it means it's going to lose its shape and it becomes biologically inactive. You've somehow changed it so it doesn't do the same thing. Sometimes it can be reversed in the lab, but generally in nature, when you denature something, you have changed it for good. It's permanently changed. So if you think of taking an egg, when you crack that egg open into the frying pan, you put enough heat into it that it changes the structure of that egg permanently. You can't unfry that egg in there. It's been denatured in there. It's still an egg. It still has the same amino acids in it. You've just changed how they hook together in there with it. Some other things that can denature things other than temperature would be acidity or basic conditions outside your normal pH in there can do that to change that a lot. That's probably the more common thing, and then temperature. So when you think about that, when they talk about not letting a person's body temperature get over a certain degree, that's the reason why they say don't let somebody's temperature get over 105. Up until that point, you can usually let a fever go, but at 105 is when you're going to start to denature proteins in the body, and you can have serious damage occur in there. So the chaperonins, these are chaperone proteins that assist in proper folding. They help keep away interfering influences. So you can kind of think of it, if you're making your little origami protein animal here, the chaperonin is kind of the person that sits there and keeps the little kids that want to fold your paper for you and spill on it and everybody else out of the way. It kind of guards the situation and helps guide you. It gives you the instructions and keeps the interfering things away in there. So how can we determine these shapes of things? When you look at proteins, they literally look like you took a string of something and you wadded it up and went like this and made a glob in there. It looks like it's random, but it's not. So what they've been able to use is X-ray crystallography to determine the 3D protein structure in there. Of course, you know, yes, it's using an X-ray in there, but you're having to magnify it a whole lot because, of course, it's way too small for the naked eye to see. Nuclear magnetic resonance spectroscopy is a new method that does not require forming crystals in there that will do that. And we've been able to look at enough of these things to be able to make predictions using computers. And that's what bioinformatics is using computer predictions to predict the structure of an amino acid. So that's actually a good place to stop today.